So is Dr. Ortigas back?
This is why I said 500%.
Okay, why don't we go ahead and get started today? Just a couple of brief announcements. If I can have your attention, please. We are having the poster construction workshop today at 4 p.m. over in the library. Does everybody know where to go? You know where the Teaching and Learning Center is? Or find a group member or ask me at the break if you don't know where it is, but it's in the main library on the first floor. So we'll meet over there around 4. There is another seminar that's being held in that same room, which I'm actually attending myself. So it won't start until almost exactly 4, a little bit after. So you can come into that area and just be sure that you wait till all those other people leave before you pile in. There's about 36 workstations, and so that means not everybody can go, but most of you will have access to a computer, so you can work along with Dr. Ching Lee, who's going to be directing the workshop. And I'm going to be there, obviously. So that's at 4, and I've moved the deadline for ranking each people in your group because I forgot to put the graduate students on there. So that's now due Friday at 11:30. Let's hope you're not working 11:30 at night on Friday and you're doing something better than that. But that does give you a little extra time to complete that. And obviously the initial contribution I think is due, well, the on-time initial contribution is due that same time at Friday. I will accept I will accept late papers or contributions, but you'll lose points for doing that. So try to get it in by Friday at 11:30. Okay, any questions about what's going on? Remember, we moved the quiz 3 until a week from today. So the material stopped with Tuesday's lecture. So all that protein, the whole test is on one topic, which is large, but it's protein targeting. Okay, so that's the whole test. Well, except that it is comprehensive, isn't it? Okay. All right, you did, Kyle. All right. Okay, so we're going to start a new topic, and this is a really broad topic. This chapter is massive. The way that they describe signal transduction is to give you a bunch of theoretical, no examples, just here's some little boxes that move around, and I really don't like that style. So when I do things, I like to give you particular examples of things that are going on. So in the reading, you'll see that I have you skip right to some of the stuff that we'll start with. So we will so this is the brief general scheme of the signal transduction scheme. And so you should have had that in introductory biology, but we will go through it briefly. And because you need to be able to describe those basic pieces no matter what signal transduction pathway. We won't get to, obviously, every single signal transduction pathway that's in your book, and your book doesn't cover even a third of what the actual numbers of signal transduction pathways there are. So we'll use very specific poster children as our examples and in some detail. And some of your projects that you've discussed with me, and I appreciate those people that have done that, it's clear that if you're working on inflammation, you're going to need to know the NF-kappa B signaling pathway. We won't get to it, but it's in your book. And if you can understand how these basic systems work, you should be able to decipher any signal transduction scheme. For example, Michael Dean, that gave the cancer talk, he talked a little bit about the disheveled and smoothened pathway. And that pathway's in your book. We just won't have time to get to it. So you should be able to read that and understand it on your own if you understand the basic components of any signal transduction pathway. So let's do that visually, okay? And just to start out with, the way most signaling systems worked was when unicellular creatures started to communicate with each other. That had to precede the complicated signal patterns that we have in our cells. So it took 1 billion years for a life to rise on the planet. It took another 1.5 billion years for multicellular organisms to show up. Why the big difference? So why did it take 2.5 billion years for something as complicated as a multicellular organism to show up? What's an example of that? Or why do you think that's the case? Well, how many cells do we have? 100 trillion. And that's just the eukaryotic cells. We have even more prokaryotic cells invading us all the time. But in order to have a complex organism work with multiple cell types, they have to communicate. So some people believe it was actually the evolution of these complex signaling patterns that had to occur first before you could get a really organized multicellular organism to come together. Okay, so here we have, and that began with unicellular organisms talking to each other. This is yeast. Okay, and when they're not excited, they're round, spherical things. But then when some of them secrete a mating hormone into the extracellular space, they respond by getting a yeast erection. Okay, so nothing worse than a bunch of horny yeast floating around. Okay, but this is the basis of responding to extracellular chemicals, and then those cells were able to form part of a multicellular organism and talk to one another, and so it had to be preceded by some sort of signal and some way to receive the signal, and so we have this general scheme that looks something like this. You have, and this is for a signaling system that is hydrophilic. So if the signal is dissolvable in water, that makes it difficult to go inside the cell and act on the cell. So a lot of signals actually never make it inside of the cell. Okay, they do the signaling by that little red dot. It could be a hormone, it could be growth hormone or something like that. It binds to a receptor. The receptor's job is to change its shape in response to binding to the hormone. That's its main function. Okay, but it also can lead to the generation of a new signal. So we call that signal, that's called transduction, when an extracellular signal is converting to an intracellular signal. So one of the jobs of the signal transduction pathway is to turn it into an intracellular signal. The second function is to take that signal and amplify it, because you can start with a really tiny level of signal on the outside, and you can end up with an explosive change in second messengers, as you will see. Okay? And what do those second messengers do? They instruct the cell how to respond. So, for example, you have a typical second messenger is cyclic AMP. We'll talk about what that is. So you take ATP and you split it and you link the 5' and 3' hydroxyl groups together by using a phosphate bridge, and that's cyclic AMP. And then that has to bind to various proteins that are called effectors. So the second messenger activates effectors. And generally, effectors are protein kinases. That's a really good example. There are other effectors that we'll cover, but that's a really basic idea. The intracellular message activates some sort of protein kinase, and they go along phosphorylating or dephosphorylating all the target effector proteins, so metabolic enzymes such as those that break down glycogen. Obviously, you're going to talk about activating different transcription factors that come and change gene expression. So we'll be linking those two ideas that we started at the beginning of the course. And of course, you can actually make the cell change its shape and how it migrates depending on what happens to the cytoskeleton, which we'll cover in about a week or two. Okay, so that's the general scheme. Let's get into the specifics. How do you categorize these different signaling pathways? Well, the most fundamental way is to categorize them by the type of receptor that they use. That's how you categorize signal transduction pathways. And we're going to start with one of the largest families. It's called the G protein coupled receptors. And so we need to understand what a G protein is, but we also need to understand what kind of receptor is responsive to those signals that are coming from the outside. Lots of hormone receptors are G protein coupled receptors, okay? And so we have almost over 700 in the human genome. They're all over our nervous system. They're in our metabolic organs. They're just all over the place, okay? And so the ligands can be really diverse, ranging from light, small chemicals that we detect as odors, peptide hormones, or neurotransmitters. So they're all G protein coupled receptors. In fact, the pharmacological industry, the main target of most drugs is a G protein coupled receptor. So that's why they're medically important. Okay. So we need to go through how do you identify a G protein coupled receptor? What does it look like? And how does the signaling pathway take place? The first thing we have to answer is how do we generate a second messenger by activating the receptor? So we have to link up those two ideas, and we will do that. So let's just get a couple of pieces under our belt before we get back to this complicated picture. Okay, so here's going to be the first example we'll use, and if you want to have a specific example of a ligand that's going to activate the system, you can think of epinephrine or glucagon. Okay, epinephrine or glucagon. What does epinephrine do to your heart? What does epinephrine do? Why do you give people that are dying of heart disease epinephrine? You push. Yeah, it increases the rate of contraction and the strength of contraction. Okay, so, and we actually talked a little bit about how you can do that with different drugs already, with the calcium, sodium, antichlor, and fooling around with the sodium pump. You can make it more contractile. But a better way that your body does is to take a sympathetic neuron that's innervating the heart and dump epinephrine onto those cardiac cells. Okay, so that's going to be our goal for that poster child, is how do we actually make the heart beat faster by dumping a hormone. And epinephrine comes from a couple of places. So norepinephrine is a slightly different molecule coming from your sympathetic nerves. And then when you get all excited or you're scared, epinephrine is secreted by an endocrine tissue called your adrenal medulla. Okay? And so both of those types of signals are going to do the same thing. They're going to activate a G protein coupled receptor. So that's what this little guy is here. Okay? Then we have an inactive G protein, and that G protein stands for the fact that it contains a GTPase. You already know how those GTPase switches work, right? Because we've talked about dozens and dozens of those, and I'll give you a brief look at how this type of G protein is different from RAS and the eukaryotic initiation factors and dynamin and RABs. Those are all the ones that we talked about. Those are all very small monomeric. These are they're heterotrimers, okay, heterotrimeric G protein. So it's got 3 different pieces to it, subunits. And then this is our signal transducing enzyme that's going to generate the second messenger, which is going to be cyclic AMP in the system. So somehow we have to get all these things to talk to each other. And I really like your books. Your book has a good video. I think it actually works on your DVD, but let's take a look at this one. And because it's a fairly decent explanation. And then we'll go back and hit the components again. So I want to turn this down just a bit. This, and I'm going to...
When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions.
Okay. Before that goes very far, the way you can identify a G protein coupled receptor is extremely easy. They're called 7-helix receptors for a reason. They always have an N-terminus that faces the outside, okay, and they have 7 transmembrane alpha helices. And if it's got those, it's 9 times out of 10, it's going to be a G protein coupled receptor. The C-terminus, of course, has to be on the opposite side of the membrane if it's got 7 odd number of alpha helices. And so this exposed site is going to be where it binds the ligand. The inside is undergoing a conformational change that's induced by what? Ligand binding. That's the only thing this receptor is going to do. Okay.
Which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade. Eventually, the alpha subunit hydrolyzes its bound GTP to GDP, which inactivates the subunit. This step is often accelerated by the binding of another protein, called a regulator of G protein signaling, or RGS. The inactivated GDP-bound alpha subunit now reforms an inactive G protein with a beta-gamma complex, turning off other downstream events. As long as the signaling receptor remains stimulated, it can continue to activate G proteins. Upon prolonged stimulation, however, the receptors eventually inactivate, even if their activating ligands remain bound. In this case, a receptor kinase phosphorylates the cytosolic portions of the activated receptor. Once a receptor has been phosphorylated in this way, it binds with high affinity to an arrestin protein, which inactivates the receptor by preventing its interaction with G proteins. Arrestins also act as adapter proteins and recruit the phosphorylated receptors to clathrin-coated pits, from where the receptors are endocytosed. And afterwards, they can either be degraded in lysosomes or activate new signaling pathways.
Okay, that was a mouthful. That's pretty much the whole story for G protein. So let's step back and see if we can go through this. And the details of those steps are all written down here in the outline. So what we're going to do is just step through them visually. And so let's go back to the PowerPoint. I'm sorry? The what? The comment? Oh, yeah, YouTube. Yeah, you never know what you're going to get on YouTube. I have to be careful about that. You take that, you dirty GT. You dirty rats. Okay. Okay, so there's the components, and this is an old story you're familiar with. What is the GEF in this system? You saw what happened. The hormone, let's take epinephrine, found that green thing, okay, activated it. What did it do? It bound to what? The heterotrimeric G protein. So what is the role of an activated receptor? It's a GEF. That's all it is. It's a GEF. You already know the story, okay? So that plays a role as the GEF, and it bumps into the heterotrimeric G protein. It's the alpha subunit that is the GTPase in this picture. We'll talk about what the beta gamma subunit does, but that alpha subunit is not only the GTPase, so it's going to do this little cycle right here, okay? But it's got its own gap. It has its own, so it has two domains. It has a RAS domain, because RAS is a monomeric GTPase, right? But RAS cannot split its GTP unless a gap protein bumps into it and tells it to do something. Okay? This one has its own gap domain that after a period of time will induce it to split its own GTP. So it's got its own off switch. Right? So it bumps into a GEF. That GEF is the hormone activated receptor. And it's certain domains in that receptor that have changed shape, right? So here's the blobby green thing, okay? And so it has a couple of intracellular loops. This is C2, this is C3, and then this is the C terminus. And so these little loops here are the things that interact with the heterotrimeric G protein. Okay? And they cannot interact with the G protein to activate it unless it's bound to the ligand at the surface. Okay? Now here's the rule. Large ligands like, let's say, pituitary hormones, LH and FSH, those are big 200 amino acid proteins, basically. Those ligands actually bind to the N-terminal domain that's much larger than most G protein coupled receptors. So if it's a large ligand, it will have a large G protein coupled receptor with a large N-terminus. And so it doesn't get down in here and bind to these little loops here. But if it's a small molecule like epinephrine, which your book, being British, calls adrenaline, that's the wrong name. It's epinephrine is the term. Okay, that epinephrine will actually burrow down into the exoplasmic leaflet and work its way in here. So it gets right down in there. But it's still the extracellular domain that the ligand is talking to. But what that does is it induces these parts of the protein to change shape. And they recruit the G protein to it. And then this thing acts as the GEF, right? So what's going to happen is this G protein, this is in the inactive state, and you know why? It's got GDP, right? You got to get rid of the GDP. So the point is to bind to this with a hormone activated receptor, and it's going to mainly interact with the alpha subunit, and the alpha subunit and the beta gamma subunit are always in the plasma membrane. And there's a good reason for that. All of its binding partners are also at the cell membrane. In fact, if you have one of these G proteins that's causing cancer, one way to prevent that protein from causing cancer is to cut off the lipid anchor, right? And if it can't work with the plasma membrane, it can't work. This is not a retractable tail, okay? It's not like the RAB system where it can raise and lower its landing gear. The landing gear is always deployed. Okay? And there's two pieces that have the landing gear. It's the alpha subunit. The beta subunit is an indirect peripheral membrane protein. Okay? It's a Klingon, right? It's always bound to the gamma subunit, which has its own lipid anchor tail. So that's the construction of this thing. In the off state, they're all stuck together. So the role of the hormone-activated receptor is actually to cause this to lose GDP and pick up GTP. And what that GTP does is it brings two little domains, they're called switch domains, and those negative charges that protrude, they bring the two switches together. And when they're in that shape, it can then go talk to some effector protein, which is the next step. Okay? So what happens is this becomes activated by binding to a ligand. So here it is. This changes shape, showing by those little lightning bolts there. Okay, it interacts with the heterotrimeric G protein. This loses its GDP. It picks up GTP. Now, typically, what happens is there's two dissociation events. Okay, not including the GTP. What happens is generally the alpha subunit will dissociate both from the activated receptor, number one, and the beta gamma subunit will disassociate and actually go activate its own targets. So the beta gamma is quite capable of activating targets, and so is the alpha subunit, as long as that remains in the GTP state. Right? And the reason it's really important that this dissociate is so that this activated receptor can go around, as you noticed in that video, and it activates other G protein coupled activates other G proteins. So this activated receptor right here could probably turn on about 100 different G proteins, and each one of those are going to separate like this and do its job. Now, more recently, we've discovered that these two pieces, the alpha and the beta gamma, do not always completely separate. In some cases, they stay weakly stuck together, and as a group, they talk to a transduction protein to turn it on or off. Okay. All right. So let's superimpose this knowledge onto a specific example. Let's say we have a cardiac muscle cell. You dump, so you get scared or excited, or you're playing athletics or whatever, and so your adrenal medulla secretes epinephrine into the bloodstream, so therefore it's a hormone. That hormone is going to bind to it's called the adrenergic receptor. The reason it's called that is because of the British system, adrenaline. So if adrenaline binds to that receptor, it's called an adrenergic receptor. That adrenergic receptor is then activated, and this heterotrimeric G protein complex is then activated and does its job. All right. Following the picture a little bit further, what's going to happen is the activated G protein is going to talk to some sort of transduction protein. That is, it needs to transduce the outside signal into an inside signal. And that's the job of this little thing here. It's completely misdrawn. It's actually, this is adenylate cyclase, or it's actually called formally adenylyl, but it's hard to say, so people say adenylate cyclase. And what it's going to do is it's going to take ATP as the substrate and turn it into cyclic AMP. Okay? So you're going to get a little burst of cyclic AMP. Do you also see some amplification here? We've already seen a couple of steps where this signal has been amplified. Number one, this activated receptor can can talk to more than one G protein. Right? I said it can talk to about 100 different ones. So now you have 100 activated G proteins. Okay, so that's one means of amplifying it. This thing is an enzyme, so it doesn't take one ATP and make it cyclic ATP. It takes 1,000. So you've now exploded the signal times 100 times 1,000, so it's 100 times 100,000. So it's a large signal that's ballooning. Okay. That cyclic AMP is then going to bind to some effector enzyme. In this case, because we're talking about cyclic AMP, we call that protein kinase A for cyclic AMP. That's where it gets its name. And this is kind of an interesting protein itself. Okay, this is just showing what's going on. You don't really need to know the chemistry, except this is the starting point, and what you're doing is you clip off a pyrophosphate, which is highly energetically favorable, and then you retain one of those phosphates and you make a phosphodiester bridge, just like you would in DNA. But it's between the 5' and the 3' hydroxyl groups. Okay? So we have a cyclic molecule. And then if we want to extinguish this reaction, there's another enzyme that's always present. It's called cyclic AMP phosphodiesterase. That's an off signal that we'll talk about here in a moment. But all you have to do is break that chain, and then it becomes an inactive signal. Okay. So let's focus on the downstream events. Now we have pKa. When it's inactivated, you have 4 different subunits. 2 are regulatory, and these 2 regulatory subunits are stuck together by disulfide bridges, so they're never going to come apart. Okay? But the binding to the catalytic subunit inactivates the catalytic subunit in the absence of cyclic AMP. So with no cyclic AMP, it's off, and it's a heterotetramer. Correct? Okay? Cyclic AMP, 4 molecules of cyclic AMP are going to bind 2 to each one of the regulatory subunits. And what that does is it changes the shape and it boots out the catalytic subunits. Okay. So those catalytic subunits are now active protein kinases, and so they can phosphorylate, in the case of cardiac muscle, they phosphorylate calcium channels, open up the calcium channel, and the calcium pours in, and what does that do to a cardiac muscle cell? That. Okay, so it's going to grip it. And so it's going to contract. And also, though, these protein kinases, if they're in the monomeric state, they can go in through the nuclear pore into that cell and replenish the supply of contractile fibers and all kinds of genes. So the way that works is it goes through, it exposes, obviously, in the monomeric state, a nuclear localization sequence, and goes through that process that you now know extremely well, or you will by Thursday. And once it comes in, now let's zoom in on this a little bit. Okay, you know this part of the story. There's the target gene. Let's say you're going to make more myosin. Myosin is a contractile fiber. Okay? And so that's the gene that needs to be turned on. And the response element is the piece of DNA that in this case would be an activator. Okay, it's an activator. And what's going to happen, it's called cyclic AMP response element, because the thing that it's responding to is cyclic AMP. Okay? But cyclic AMP doesn't bind to the DNA. What binds? pKa. So pKa comes over here and, pKa activates this protein called CREB. So it's cyclic AMP response element binding protein. So that's why we call it CREB. We don't want to say that mouthful, right? So this thing will not bind or become activated until pKa phosphorylates it. So that gives it a license to bind to this response element. So that the little green CREB binds to the DNA, and then because it's phosphorylated, it's going to recruit another protein, which is cyclic AMP response element binding protein, which we call, thankfully, CBP. So this is going to bind. This is a coactivator. It's a coactivator. Because you notice, it's not binding to DNA. It has no DNA binding domain. CREB has the binding domain. Okay. So we recruit this, and what this thing does is it sticks out its hands and uses protein-protein interactions to recruit something that's going to assemble the general transcription apparatus at the promoter. And that something in this case happens to be a HAT, a histone acetyl transfer. So it loads up the nucleosomes full of acetyl groups, and that opens the DNA, and then that exposes the promoter, and then the general transcription factors can be recruited, and you form this big collection of proteins called an enhancosome that turns on the DNA, and you know the rest of the story. So you synthesize the gene for myosin 2. And that keeps your heart structurally intact. Meantime, you have a much faster response, right? The nice thing about protein kinases is that they can phosphorylate and turn enzymes and channels on and off extremely fast. Because if you had to wait for transcription to run your heart, you'd be dead. You need something very quick. Okay? So the nice thing is you have cytoplasmic events like phosphorylate or activating channels, but you also have these slow events that replenish the heart's structural integrity. Okay, so that was a long path, but it all started with an extracellular ligand down to its receptor. The signal became amplified because you generated a second messenger that was generating an enzymatic response. If you turned on adenylate cyclase, you got an explosion of cyclic AMP. That turned on and released the catalytic subunits. Again, another effector, which is protein kinase A. It did some things in the cytoplasm. Mainly, it activated open calcium channels, but then it went into the nucleus and started turning genes on to keep that heart muscle strong and constructive. So transcriptional and then very rapid non-transcriptional effects are the result. Okay, and your heart, of course, even if you're not scared or frightened or anything, of course your adrenal medulla may be turned off, but you have to keep a heartbeat, right? So what happens is you're dumping a different hormone. It's not a hormone, it's a neurotransmitter. It's norepinephrine. It has a slightly different structure than epinephrine. It's got one methyl group different. Okay? Okay, questions?
What subunit of the G protein did you say went around and activated all the G proteins?
It's not a subunit of the G protein that activates other G proteins, it's the activated receptor.
Oh, okay. I thought you said that once the G protein associated because of the receptor, though, the alpha from the receptor and the alpha from the beta gamma complex, one of those went around the cell and activated others. But
Not G proteins, their respective targets. And in this case, the beta gamma really doesn't do anything, but I'm going to give you an example here in a moment where you do use the beta gamma. It's where they blow apart. The alpha is doing all the work in this picture. The beta gamma is not doing anything, but that's not always the case. In fact, the way we slow the heart down is a great example of when we use the beta gamma. Okay. All right, so we've gone through a lot of stuff, and so I mentioned early on, there's lots of diseases that have to do with G proteins that are malfunctional, the receptors are malfunctioning. In one particular case, which is male precocious puberty that we talk a little bit about in endocrinology. So you have a pituitary hormone that's coming out of your pituitary going into the bloodstream. In men, it binds to the testes, particularly the Leydig cell, which causes testosterone secretion. You don't want that to happen until a boy reaches puberty, and so that should be 11 or 12. It's actually occurring earlier. But in some cases, the LH receptor no longer, it's mutated, so it no longer needs the ligand. So in these children, they become precociously sexually developed because that G protein coupled receptor is on all the time. Okay, and so we can deal with that by using inhibitors, but there's lots and lots of examples of where you mess with this and you really screw up a lot of things. Okay, in general, so I didn't go through the specifics. I don't need to. It's explained in your notes, but there's large families, and we divide them into different groups. The small GTPases include examples of things that we've talked about over and over and over: RAN, RAS, RAB, SAR, and ARF. The key thing here is, how do they differ from heterotrimeric G proteins? Number one, they're called small because they're monomeric. They have no binding partners. In other words, they have no subunits. It's only an alpha subunit. That's the way you can think of them. It's like they represent the alpha subunit of the heterotrimeric G protein. The second difference, so they're small, and it's just an alpha subunit. They have no gap domain, so they must bind to a gap. They must bind to a gap. That pushes the subunit, this GTPase subunit, to split GTP and extinguish the signal. Okay. In a heterotrimeric G protein, what happens is it's quite capable, if you leave it alone, it takes about a minute, and it will activate its own gap domain, and that causes GTP to hydrolyze back to GDP. And what happens? That changes the shape of the switches. They fall apart. Okay, so the alpha subunit is turned off, and then it cannot be reactivated until it rebinds to the beta gamma subunit. So it has to come back together to form a heterotrimeric off complex, and then it can be reactivated. But an alpha subunit bound to GDP cannot be activated without its binding partners. It must be bound to beta gamma to be reactivated. Okay. So most of these families are small GTPases, and then the one that we're focusing on here are the G proteins, and we've talked about all the different components in that cycle. Okay. All right, now the video mentioned something. So there's 3 different ways to turn off the alpha subunit. Okay. Number one, it's quite capable of turning itself off. It has an intrinsic gap domain, okay? But it takes a little while. It takes a minute. That's a long time in molecular terms, okay? If we want to push this a little faster, we use RGS proteins, which stands for regulators of G protein signaling. What are they? They're gaps. So they do have gaps. They just don't need them. But they will accelerate and cause better, more exquisite control over the G protein signal. So there's quite a few of them. There's maybe at least 25, probably more now in the human genome. So these things are floating around. They're actually anchored into the membrane, and they're moving around. And if they bump into the alpha subunit, it will cause it to activate its GTPase, and then it will turn off. So that's number 2. And then when it talks to the effector, which in this case is adenylate cyclase, just touching the receptor causes the gap domain to activate after a delay. Okay, so when it's touching, it's activated, its effector, that's actually slowly going to turn on the gap domain, and it will split GDP. So these are all safety switches, because you don't want to leave a G protein on for very long. You want to get your thing done, and then you want to extinguish the signal. Now, you noticed, what's the other way that you can turn this pathway on? If that ligand, as long as that ligand stays attached to the protein, it's going around activating G proteins. So how can you stop that receptor? Well, one way is that the ligand dissociates. That will turn it off. Okay? But even if it's still on the receptor, then we have a whole class of controls. I think they're well, we'll get to them, but they're called Greeks. That doesn't stand for any kind of fraternity. It's G protein coupled receptor kinase. It's GRK, Greeks. Okay. And so what happens is those Greeks can phosphorylate. Let's see if I have an example here. No, it's not. It can phosphorylate that intracellular domain, and if you put phosphates there, that makes it very difficult for the G protein to talk to the receptor. That's number one. The second thing is other proteins, adapter proteins, like arrestin,
and bind. If you look at that video again, you'll see that. And it binds to this domain, and what that does is number one, it blocks the signaling pathway. So G proteins can't get past the arresting protein. And we'll talk about this in more detail later, but it also recruits the clathrin mechanism to pull it off the cell surface. That's called desensitization, when you actually remove the receptor. And now you know what you can do. There's two different fates, right? You can pull that receptor off, send it to the endosome, right? The early endosome, and then you've got two choices. If you want to destroy it, where do you send it? Late endosome. And then that says you have to go to the lysosome, right? And that's what happens to the opiate receptor. And sometimes it happens to this, but in many cases, if you want to keep the heart beating really fast, you're in a particular situation, okay? What it does is it recycles the receptor back to the cell surface, so you would go from the early endosome to the recycling endosome and then back to the cell surface. So all these pieces of the course are starting to come together, hopefully. Okay, so now we're going to shift our focus a little bit to the actual signal examples of second messengers. So we talked about one, and that's cyclic AMP. So let's just ask the question in general, what is it that we're looking for? What actually makes a second messenger a good messenger? And there's things that you have to consider about that second messenger. Number one is the changes in concentration is one part of the signal. So it's carrying information, but it's encoded in several different ways. Number one is the concentration. Okay, how high is the amplitude of the signal? Number two, what's the pulse frequency? So what happens, it turns out you just don't turn on this big blob and it stays up there. It comes in pulses. So the pulse frequency of the signal is carrying information. If you get rapid pulses, you're going to really hyperactivate the system. You get slow pulses, you get a different response. Okay? So we have proteins in the signaling pathway that can decode the pulse frequency. Making this really complicated is you never have just one pathway activated. There's usually multiple messengers talking to the cell at once. And it's the integration of all those different messengers that tells the cell exactly what to do. This is a general theme in cell biology. It's the same thing about combinatorial control over gene transcription. It's the same players, but unique combinations make the message. And that was true for targeting of proteins to their destination. Unique combination of coding proteins and targeting and fusion proteins. So it's a unique combination. And here, it's a unique combination of multiple messages. Okay. And then where the location is very important about where the response happens. So these signaling molecules tend to be somehow associated with the plasma membrane, but also within the cell, there's a way to spatially localize those signals so that they act much more faster. So a good example is with cardiac muscle. So let's talk about that briefly, and then it's time for a break. Okay. Let's just do this one thing though. All right. So back to the heart example. We've got a cardiac muscle cell. Okay. And we've got a heteroprimary, G protein coupling receptor, 1, 2, 3, 4, 5, 6, 7. Okay. This is the beta. Is this in focus? Yes. Okay. Adrenergic receptor. And this word right here tells you that it binds to adrenaline or adrenergic. Okay. And so this is a specific example of a G protein coupled receptor. GPCR. That's the generic term. G protein coupled receptor. All right. And so what's going to happen is epinephrine is going to bind to this, okay? And then it's going to recruit a very specific type of G protein. And the G proteins are named in two ways.
the way you can always tell which subunit you're dealing with because it's a G sub alpha tells you that it's the alpha subunit. And the other possibility is a G beta gamma. Remember, those two always stick together. So you don't ever talk about G beta or G gamma. You talk about G beta gamma. Okay. And then you give it another subscript, which is tells you exactly which type of alpha subunit it is. And S was the very first one. And that little S stands for stimulatory. And that's because the very first one that was found was the one that elevates cyclic AMP. So they initially thought that all G proteins elevate cyclic AMP, but they found differently. So they had to start giving the name. But this is the one that becomes active in the heart in response to epinephrine. Okay, so you know the story. I'm not even going to draw the beta gamma. You know the beta gamma is then going to dissociate. This is the GEF, and so this thing loses GDP. It picks up GTP, right? So it's now GTP bound. And that state, it can then talk to big AC. It's not air conditioning, it's the adenylate cyclase. The reason that little cartoon in your book is so irritatingly inaccurate, it has 12 membrane spanning alpha helices. It's not one, it's 12. Okay? So it's an honest to goodness membrane protein. Okay? And so that's going to take what? ATP and create cyclic AMP. So there's your second messenger. And then the second messenger, now here's where the localization becomes important. Because what happens is this is going to activate PKA, and we want a very fast response. Of course, it can go into the nucleus and do all those wonderful things with Krebs and CDP and all that stuff. But you would die after that did anything to your heart. Okay. So what there is it's going to bind to PKA, and then there is a mechanism by which this becomes localized to the plasma membrane. So we're going to put a protein here, which is called ACAP. ACAP. And what that stands for is A kinase anchoring protein. And so there's all kinds of anchoring proteins for all different kinds of enzymes. And in this case, you want the PKA to be localized to the plasma membrane. So where do you put the ACAP? It's not any ACAP, it's ACAP 15. It's in the plasma membrane, right next to calcium channels. Okay? So the ACAP is going to recruit the PKA to it. So the PKA is now sitting right here, and it can put a phosphate group onto the calcium channel, and then that gates the calcium channel open. And so calcium is going to pour into the cell and make that cardiac muscle cell contract. But it all starts with components that we're already familiar with. A G protein coupled receptor turns on a G protein. In this case, we have to start being specific because different isoforms of different G proteins do different things. G sub S, alpha, is very good at turning on adenylate cyclase. And so that part is all very standard. AC makes ATP, or splits ATP, makes cyclic AMP, activates the PKA, and then that's recruited to the plasma membrane to do its job on the calcium channel, which is gated by phosphorylation. And then your heart contracts. And when we come back from the break, I'll tell you how to stop that contraction. Yeah. Adenylate cyclase. AC is adenylate cyclase.
Dr. Collar, did you come?
Oh, gee, this is bad. Okay.
We'll be back that night. I can take it
Okay, that would probably be best. Yeah, so you don't have class Friday? Why don't we say,
I need to make it earlier.
okay, oh, yeah, okay. We'll just, I'll reserve. Probably, if I'm not there, what I'll do is I'll listen to well, she does this this Let me. Yeah,
Well, the last courses that I have, sometimes it's last night. Exactly, that's right.
okay. We're gonna talk about it. Okay. Well, one way you could do it is the way it's done now. Is they take their huge you can put different compounds in different wells. Look at thousands. You know which one. And every well has you can put your stats somewhere and see if
By the way,
you don't have to do that. That's what you use as a chemical. It's selected for.
not actively.
You want it to, well, that's up to you. It's not so you just want some small stop. Oh, okay, so you need something activates. So you have a couple of those.
No, I just like do that, like looking at the different ones and like a good copy. Like I really feel like focus on afterwards, but yeah, so I mean, it kind of it just like, what do you prefer the uniform? Like, I really done my background was probably about like half of it, like economy, and then, I mean, I think that's really like, then you're also like 2 pages of and by the way, if y'all just Google Scholar, like, will cite it for you. Like, because he didn't learn how to study it, so I just properly placed it. Like, go to, like, find the article and then and it gives you the form. So you have to just see whatever you want. See what you know.
Okay, why don't we get started? We've got about 20 minutes. All right, so obviously based on what type of G protein, what alpha subunit, what beta gamma subunit as well, but we classify them by the alpha subunit. So that tends to be the major player. They do different things, okay? So, for example, as you saw, G sub S, the active component is the alpha subunit. It activates adenylate cyclase, and because of what it can do with the pKa, it activates calcium channels in some situations, okay? This was the other half of the story. Not all G proteins are activators, so that's not surprising. So the ones, in this particular case, that actually works on adenylate cyclase is called a G sub I. So the G sub I alpha is going to do something, and in this case, the G sub I beta gamma does something else. So let's look at what it does. So if you activate this guy, the alpha subunit inhibits adenylate cyclase. Now, what does that in the heart is acetylcholine. So your vagus nerve is the parasympathetic other half of the story. The sympathetic nervous system is secreting norepinephrine, which is stimulatory. You're also getting epinephrine from the adrenal medulla. So that's all the stimulatory half that's making your heart go faster. But what do you do when you want to slow it down? Well, you want to activate a receptor that's talking to this G protein. And the thing that's doing that is it's a cholinergic receptor, and we now know, we talked about this a little bit, you have nicotinic receptors. You know what those are. What do those do? What's the mechanism? Acetylcholine binds, and then the receptor is what? It's a channel. It's a nonspecific cation channel. Usually lets sodium in. That's not what's going to be activated here. So we're going to have to bind to a G protein coupled receptor, and those are called muscarinic, because muscarine, which is a compound, I think it's also from mushrooms, when it binds to the G protein coupled receptor, it binds in the acetylcholine binding site and mimics acetylcholine. Okay, so there's different flavors of muscarinic receptors. In the case of the heart, you activate a very specific one. It's called muscarinic number 2. So M2 receptors are G protein coupled. How can you tell? They have 7 alpha transmembrane expanding alpha helices. So you immediately go, well, this is coupled to some sort of G protein. So let's do a little diagram, finish our diagram here. Okay, this is way too crazy, so let's move that out of the way. So this is the same heart cell, right? So your vagus nerve says you're trying to relax, all right? So your vagus nerve is going to release acetylcholine, and acetylcholine is going to bind to the M2 receptor that I just described. So you know that means it's a G protein coupled receptor. But it's not going to talk to G sub S. It's going to recruit G sub I. So it's G sub I alpha, and this case, which gets turned on. So the alpha, the G protein is activated, but what that activated G protein does is to turn things off. What it does is when it's activated, and you know how it's activated, it loses GDP, it picks up GTP, it dissociates from the beta gamma subunit. Let's say we send that off over here. So the beta gamma goes over here, right there. The alpha goes in a different direction. So this is a good case where they have to split apart, right? And they have to dissociate from the receptor in order to do their job. So what happens is this goes over here. Here, this is adenylate cyclase again, AC. Okay. And you know what that is. That's an enzyme that makes cyclo-KMP, except this one inhibits adenylate cyclase. So what's that going to do to cyclo-KMP? It's going to drop it, right? So you're extinguishing the signal to activate pKa, which opens calcium channels. So you're turning that off, so you're going to close calcium channels. That's number one. You close the calcium channels because you're getting rid of cyclo-KMP. Okay? What's this guy doing? This one is not silent this time. It's going to do its own thing. And what the beta gamma subunit, I'm confused by my own diagram. So beta gamma, just really oh, I know what it is. It's actually in that previous slide. It opens potassium channel. Now, why isn't that going to stimulate the cardiac muscle cell? So this is a K channel. It's going to open. What happens when a K channel opens? Which way does the potassium move? Pardon? It's going to leave, right? You don't even need, you really actually do need to do the Nernst equation, but let me tell you, it leaves, okay? So it's going to leave. What does that do to the membrane potential of the cardiac muscle cell? It hyperpolarizes. What's our rule? Hyperpolarization means off. Depolarization of an excitable cell, like a cardiac muscle cell or a neuron, turns it on. So we're doing the opposite of turning it on. We're going to hyperpolarize. Polarize the cell. So this becomes much more negative. So the membrane potential decreases, becomes more negative. And it makes it hard to excite that muscle cell. And so you got to excite it to contract it. So we're making it less excitable. So we're calming it in two ways. We're turning off the cyclo-KMP with the alpha subunit, and then the beta gamma is doing its own thing to calm the cell by hyperpolarizing. So there's a lot of lessons from this. The beta gamma is not always inert. Sometimes it's doing its own little job to regulate contraction or whatever the system is. And the other thing we learned from this is that activating a G protein doesn't mean we're going to activate something. We could be using it to turn off the system. Turn off the system. Okay. Any questions about that? I mean, there's dozens and dozens of G proteins we could use, but these are going to be our main poster children. Yes? What is it doing? It's opening. Yeah, it opens. So we'll just put that up there. Opens the K channel. Yeah. Inhibiting it? No, that happens too. Because the kinetics of the way they do their jobs are different. So it's like a built-in control switch where the alpha subunit is doing this. Let's say it's stimulating. And then the beta gamma is actually doing the opposite, inhibiting. But they don't work at the same speed. So you turn on the thing, and then this starts to turn it off. If it's something that's really sensitive, it's dangerous if you leave it on, kind of like this, you know, you've got all kinds of controls. Sometimes, it's usually not
invested in the same G protein. Usually you've got different signaling pathways like you do here, converging on one cell. You have the you have epinephrine, you have acetylcholine talking to it, and then there's dozens of other things, atrial, natriuretic peptide, there's all kinds of things talking to it. But there are a few cases where the alpha subunit is doing one thing, the beta gamma is doing an opposite reaction. It's pretty complicated. Okay, let's go move away from that. So, to summarize that, G alpha stimulates AC, some beta gamma stimulate, others inhibit AC. A good example of, so we talked about the way beta gamma does its thing, but we're not going to talk about every particular situation. This is the G sub alpha. You can actually add something to this now. It inhibits AC, but it also activates K channels, and that's what causes the cell to hyperpolarize. And again, just like for transport and for protein trafficking, we've got some tools to help us study these things. And one of those things is forskolin, okay, which is a plant chemical, and that directly activates adenylate cyclase. So if you have a system and you don't want to look at the influence of the receptor or the G protein, you just want to turn it on and see what happens downstream from that signaling pathway, you can dump forskolin on that cell, and if it's got adenylate cyclase, it will make cyclic AMP. So it's a way of sort of punching it and making sure it works. All right, so we've talked about cyclic AMP, and then this one's a really simple one. This should make a lot of sense to you. It's cyclic GMP. Okay, so you start out with GTP, and then you split off a pyrophosphate, and you make cyclic GMP. So it's just a different nucleotide that binds to, surprise, different effectors, right? So one of the interesting effector systems in this is guanylyl cyclases. So it doesn't activate adenylyl cyclases like cyclic AMP does. It activates different cyclases. And those cyclases end up being located in different places. There's some in the cell membrane. This is the one we're going to talk about. The guanylyl cyclase is actually in the cytoplasm. Okay, so let's look at a system where you activate cyclic GMP as your second messenger. And so to do that, we're going to have to look at some different, some new ligands. And your book has a pretty good example of that. We've gone through all of this. Okay, so this is cyclic GMP. So that's the second messenger. And obviously you need a guanylate cyclase to produce that. But this is the system in which that type of action occurs. So we have, this is an arteriole. So this is something that controls blood pressure and blood flow and different types of organs. So this is the lumen of the blood vessel. And the vessel is actually constructed of a single layer of endothelial cells. So that's the wall of the blood vessel. And it needs to be really thin, not only for gas exchange, but because of these cells are going to be talking to one another. So the inner lining is where the blood is going through. And so if you squeeze it, what's going to happen? Decrease flow. If you dilate it, you're going to increase flow. Okay? So what's controlling that is this little machine right here. These are vascular smooth muscle cells. And so you can control the diameter of this thing by how much squeeze you put onto it. If you relax it, it opens up. Endothelial cell gets bigger, the blood vessel diameter gets bigger, you get more blood flow. And the opposite thing happens. This is controlled by nerves, okay, in many cases, and they tend to be cholinergic nerves, so they're releasing our old friend acetylcholine. So you have this situation where the endothelial cell is the target tissue that's actually responding to the neurotransmitter. So you saw that nerve in the previous section. That nerve is dumping acetylcholine, but it's actually migrating through here in this situation and binding to the endothelial cell. Right? You get a different response because of that. So what happens is that acetylcholine talks to a muscarinic receptor that your book doesn't tell you what it is. And I can't tell you what it is until next time because we haven't covered the type of signaling that's involved. So it keeps this a mystery. In fact, your book never tells you how this, I don't know why the big mystery is, but anyway, we'll talk about this little piece next time when we get some more players. But what happens is an enzyme is activated. It's called nitric oxide synthase. And some of you actually are looking at some projects that activate this one. So NOS is nitric oxide synthase, and what that enzyme does is kind of interesting. It takes amino acid arginine and creates a small gas, nitric oxide. And this is not the laughing gas in your dental office. It's not nitrous. This is nitric oxide. And what's cool about this is it allows for really close communication between two adjacent cells by the diffusion of a gas. So that nitric oxide doesn't have to be transported. It just goes next door. Actually makes that sound. It goes right next door to the smooth muscle cell. And it goes into the smooth muscle cell, and that's good because it's in the cytoplasm is where the target is. That's where guanylate cyclase is. It's sitting there. This is an intracellular guanylyl cyclase. Okay? So that actually binds to the guanylyl cyclase and causes GTP to be cyclized. Okay? So it makes cyclic GMP, and then it hides the answer from you about what's going on. So what happens is, the only thing you need to know is that cyclic AMP is going to bind to, what's your guess? What did cyclic AMP bind to as its effector, both for transcription and for the calcium channels? PKA, okay? This is PKG, protein kinase G, okay? So protein kinase G phosphorylates the smooth muscle cell machinery in such a way that it relaxes it. It turns it off. Okay? And so what happens in response is that the smooth muscle cell is going to relax, so it's going to get allowed to get bigger, more blood can flow through there, and you get more blood flow, right? Okay. Now, the sort of the end of the story to this ends with a drug, okay? And that's with adenylate cyclase, obviously, goes here. And then the thing that actually extinguishes it is an enzyme that's in the cytoplasm called cyclic ANP phosphodiesterase. So if you want to turn off the cyclic ANP pathway, you can actually activate this. When we talk about calcium signaling, we'll talk about a specific example. But what if, we have about 5 minutes, so what if you want to turn off cyclic GMP? How would you do that? Well, yeah, you can imagine that if there's a cyclic ANP phosphodiesterase, there's probably a cyclic GMP phosphodiesterase. Okay. And so when you do that, what happens is the smooth muscle contraction is going to lose that nice relaxation. It's going to constrict. If this enzyme is active, because it's going to destroy the cyclic GMP signal that we generate. Right? So cyclic GMP phosphodiesterase is an off switch for this system. Now, as you can tell, people were interested in controlling this whole system to increase blood flow. They were actually interested in lowering blood pressure. And so they tested a number of drugs that acted on cyclic GMP phosphodiesterase. So it was the GMP version of this protein. And what they noticed is that the blood pressure in these volunteers, they happen to be all men, didn't do much, but they all got erections, like those yeast, you know? So they went, whoa, this is pretty bizarre. And it turned out that drug, which is Viagra, that's the way Viagra works, that's how they discovered it. It is a antagonist of cyclic GMP phosphodiesterase. Okay? And so it causes more blood flow into the penis, and you get longer erections, or you get some erection at all, or a bunch of happy yeast. I don't know what it is, but anyway, that's Viagra. And so it's an inhibitor of cyclic GMP phosphodiesterase. Okay. Got 3 minutes. Let's see. Oh, and I got to tell you my Eli Lilly joke. Some of you know what's going on here. So we had a representative from Eli Lilly come in and try to recruit students like you to go work for Eli Lilly. And
Pfizer makes Viagra, right? So he didn't want to talk about Viagra, and I said, well, what is the erectile dysfunction drug that you guys make? And they make Cialis. And so the joke that was going around the Eli Lilly institution was, if you have an erectile dysfunction, you go see Alice. She'll take care of it. So, anyway, pretty humorous guy. Okay, so just to review, we got activates protein kinase A, cyclic GMP. We've talked about all of these things, and we talked about these guys. What we're going to talk about next time is a different second messenger, which is calcium. We have just a little bit to clean up on here, but I think we'll wait till next time. And so next Tuesday's lecture, we'll start out and finish this lecture.
Okay, AC,
The air conditioning unit, yes.
right, the adenal cyclin, is that considered a protein?
Yeah, it's got 12 membrane spinning out. It's just got to be a protein.
Okay, so does that also stimulate the GTP into the CGMP? We do instead of
Well, it's a transducer protein, and so the G alpha S is stimulating adenylyl cyclase, but the act of it talking to it starts to activate.